The DNA analysis process consists of multiple steps. After a kit would be received here in our laboratory, we would work the kit. That would involve cutting any samples from it, and then those samples would then move on to the DNA analysis process. The first step involves extraction, or removing any potential DNA that may be present in that sample from the cells, and then purifying that DNA to remove any other unnecessary items within the cell. After extraction, we would then go to quantitation or quantification, which would tell us how much DNA is present in that sample. Once we know how much DNA is present, we then know how much we should target for the following steps. The next step would be amplification, which is using polymerase chain reaction to make many copies of any DNA that may be present in the sample. Once we have all these copies, we can move on to the next step, which is detection. And that involves using an instrument known as a 3500 to perform capillary electrophoresis. And that allows us to detect the DNA in the sample and to generate a visual representation of an individual's DNA profile. After we're able to generate a graphical representation of an individual's DNA or the DNA profile, we then move on to interpretation of that profile. Interpretation involves a few different steps. The first step would be determining whether that DNA profile is usable. Is there enough information present? Is there too much information present? So it's all about looking at that profile as a whole to determine if it is able to be interpreted. If you determine, yes, I am able to interpret this data, the data looks great, then we would determine, is this profile a mixture? Is it from more than one individual? Or do I think this is a single source profile, meaning it's from only one individual? We can then move on to comparisons. So that would be then comparing these evidence profiles to any known reference profiles that we have. That could be from the victim, or it could be from a subject, a person of interest in the case, to determine if those individuals are included in the evidence or if they can be excluded from the evidence samples. One example where an elimination sample may be helpful, especially when we're talking about kids, would be if there's a consensual partner or an individual who has close contact with the victim. We can use that elimination standard to eliminate any DNA that may have come from that individual and not from the suspect or subject. After the data has been interpreted, the analyst will then generate a report which will have the conclusions that the analyst has reached as far as the evidence from the kit. In the report, there are many different elements, such as information from where the kit was received, whom submitted the kit, who the kit was taken from, for example, any victim that may be involved in the case. We also may have suspects or subjects listed on the report as well if that information is available to us. After that section of the report, we would move on to our results and conclusions. Some additional items that will also be in the report would be the disposition of the evidence, basically where the evidence is now going or what will be done with it now that our analysis is complete. So that could be typically for APD, that would be just returning the evidence back to APD and ensuring that it is stored safely and securely before it's returned back. And that would also include returning back any extracts from our DNA analysis as well to APD.